Dear learners, in continuation to our quercetin, today we will be learning about another polyphenol, Camphorol. Camphorol is a natural flavanol, a class of flavonoid found in many fruits, vegetables, herbs and fruits like grapes, tomatoes, broccoli, tea and ginkgo biloba leaves. This biologically active compound exhibit many pharmacological activities including antioxidant, anti-inflammatory, antimicrobial, anti-diabetic and anti-cancer activities. The anti-cancer activity of Camphorol has been reported in cancer cells from different organs including breast, ovarian, gastric, lung, pancreatic cancer and blood cancer. Learners, the objective of this module is to understand chemistry and structure of Camphorol, sources of Camphorol, metabolism of Camphorol and health benefits. Let's now begin and understand structure and chemistry of Camphorol. Camphorol represents one of the most encountered aglycone flavonoid in the form of glycoside, tetrahydroxyflavin 1, in which 4 hydroxy groups are located at position 3, 5, 7 and 4 and it is yellow compound. Camphorol is found in various plant parts such as seeds, leaves, fruits, flowers, vegetables. Camphorol and its glycosylated derivative have been shown to possess many health benefits. Camphorol has a structure of diphenyl propane and is synthesized by condensation of 4 coumaryl CoA with the 3 molecules of malonyl CoA. Camphorol is natural flavonoid which has been isolated from delphinium, witch hazel, grapefruit and other plant extract. Camphorol is yellow crystalline solid with a melting point of 276 to 278 degrees centigrade. It is slightly soluble in water and well soluble in hot ethanol and diethyl ether. Learners, let's now understand the sources of Camphorol. Camphorol has been found in diverse families. The total average intake of flavanols and flavones in normal diet is estimated as 23 mg per day, to which Camphorol contributes approximately 17%. Common food that contain Camphorol include apple, grapes, tomato, green tea, potato, onion, broccoli, Brussels sprouts, squash, cucumber, lettuce, green beans, peach, blackberries, raspberry and spinach. The common botanical product which include and is used as medicinal plant include ginkgo biloba, moringa oleifera also contains Camphorol. Let us now understand metabolism and pharmacokinetics of Camphorol. Studies on the in vitro and in vivo pharmacokinetics of Camphorol commonly ingested as high polarity glycosides revealed that this polyphenol is poorly absorbed compared to the aglycons with intermediate polarity. Camphorol, lipophilic compound absorbed in the small intestine through passive and facilitated diffusion or active transport. The absorbed Camphorol undergo metabolic transformation to yield glucuronides and sulfoconjugates formed in the liver and small intestine by intestinal conjugation enzymes, as well as Camphorol and its glycosides are metabolized in the colon by the bacterial microflora that release aglycons and broke aglycon C3 ring to form compound such as 4-methylphenol and 4-hydroxyphenyl lactic acid that are either absorbed and can reach systemic circulation and tissue or to be excreted in feces and urine. To overcome the low bioavailability of Camphorol, its combination with quercetin increases its bioavailability, consequently improving its bioefficacy. In fact, studies prove that nanoformulation containing Camphorol will be extremely beneficial in improving their bioavailability and consequent efficacy and selectivity for mutated cell, while their effect on normal cell will be limited. Indeed, Camphorol exert protective effects in non-mutated cell, whereas it triggers apoptosis in those mutated ones. Limited data is available on effect of processing storage on Camphorol content in food. Let's now understand neuroprotective effect of Camphorol. Camphorol has neuroprotective effect and it is associated with capacity to reduce metalloproteinase activation. It is used to prevent protein tyrosine accumulation and protect against cell death, which is caused by oxidative stress. Let's now understand antioxidant property of Camphorol. Camphorol and its glycosides as well as Camphorol containing plants portray antioxidant potency both in culture and in animal models, and it has the capacity to decrease the production of free radical and other products like reactive oxygen species. Reactive oxygen species are aerobic metabolism byproduct that can induce malignant cell transformation. Thereafter, reactive oxygen species production inhibition can reverse malignant cancer cell phenotype. At submicromolar concentration, Camphorol is not only a potent scavenger of superoxide anion, hydroxy radical and peroxynitride, but it also inhibits pro-oxidant enzymes such as xanthine oxidase and activates antioxidant enzymes such as superoxide dismutase catalase and heme oxygenase 1. Anti-cancer properties of Camphorol. Possible mechanism of chemoprevention by Camphorol include inhibition of a carcinogen activating genes and enzymes enhancing carcinogen, detoxification by inducing phase 2 detoxifying genes and enzymes. It has the ability to scavenge free radical and modulate several signaling pathways leading ultimately to cancer cell apoptosis. Let's now understand the breast cancer. Estrogen receptor are important in both maintaining a normal mammary gland and in development of primary and secondary breast cancer. Camphorol has been shown to interact with estrogen receptor pathway and alter signaling in order to help slow the growth. This compound exhibit ability to lower the levels of aromatase, meaning estrogen synthetase, an enzyme responsible for biosynthesis of estrogen. Camphorol has also been found to induce apoptosis in breast cancer cell through extracellular signaling, regulated kinase activation and upregulation of p53 genes. Leukemia. Camphorol has been shown to reduce growth in promyelocytic leukemia cell through altering the cell cycle. In a gastric cancer case, control study found that consumption of Camphorol containing food was associated with reduced gastric cancer risk. Pertaining to pancreatic cancer, an 8-year study found that consumption of flavonoids, mostly Camphorol and quercetin, correlated with lower risk of pancreatic cancer among current smokers. Cardiovascular disorders. A cohort study in men showed that consumption of flavonoid containing food decreased the risk of mortality from coronary heart disease. Another cohort study showed that high intake of tea is inversely associated with myocardial infarction. Camphorol has also been shown to have protective effect on the apoptosis induced by ischemia, reperfusion of cardiac cells. Due to this, it has promising pharmacological role in preventing cardiovascular disease. Let's conclude, learners. Flavonols and flavones are flavonoid widely distributed in plant kingdom. Epidemiological studies have found a positive association between the consumption of Camphorol and quercetin containing food and a reduced risk of developing cardiovascular disease and some type of cancer. Numerous in vitro and some animal studies support a role of Camphorol and quercetin in prevention as well as treatment of these diseases such as neurodegenerative disease, infectious disease, diabetes, osteoporosis, anxiety, allergy, inflammation and pain. Additional animal and human studies and clinical trials are needed to better understand the positive and possible health effect of Camphorol and quercetin to further evaluate its potential as nutraceutical product development.